So I will try to make a little different perspective from what the excellent talk we just heard from Rachel, and because we do a parallel assessment of the Cas16 targets. So this one. All right, just to start, RNA modeling did not start in 2020. It's an old story. And in fact, it started, the first model is really the important one. It was from Michael Levitt in 1968 or 69. And in fact, we still have a base pair that in tRNA that we call the Levitt base pair, which is quite nice. And then over the years, many more RNA molecules were modeled, and sometimes with success, and they were very useful in developing this field, even to very large molecules. And we started RNA puzzles in 2011. All right, so just quick recap about RNA structure. So we talk about 2D structure, like in proteins, and or 3D structures. So the 2D structure is really where the helices are formed. Helices are Watson-Crick base pair formed. All right, so only Watson-Crick pairs, they form helices. And these are in red. And you see these yellow stretches here, and they are not indicated as forming interactions, but they do, in fact. All these bases, nucleotides, interact with each other. And that's what makes the fold possible to reach this type of compact state where all these helices assembled together in a very complementary fashion, despite the fact that every single nucleotide is negatively charged, which is still amazing. Right? So this is why I would re-emphasize the proper prediction, not only of the Watson-Crick pairs, but also of the non-Watson-Crick pairs. All right, so now RNA helices are bulky. You see the size is 23 angstrom. They are not really bendable easily. And the average length is 5 to 10, less than 10. And so they have a deep groove also here, so that you cannot really reach. And actually, because they are in this Assessor Cas15 target, if you have sharp ends, just no loops between the helices, naturally there are not many ways to assemble these helices and maintaining the covalent linkages between the phosphate groups. And so this is naturally easier to get to their proper, that's also a very good result. So all depends on what is going on in the loops and in the single-stranded regions. And even a four-way junction. This is a four-way junction. You see there's these four helices coming together. There is no variable nucleotides in between the CG pairs here. And you have 16 possibilities for coaxial stacking. So one of them is the right one. And that's the difficulty to find it. And here are shown the way you can have them parallel. You have to cross them because you have many ways. And in three-way junction, it's even worse. So three-way junction will be three helices with single-stranded region in between. And we distinguish family A, B, and C, where the third helix is perpendicular, or is down, or is up, and that's not the same. And what is going on at this junction is very critical, and is always formed by non-Watson-Crick base pairs, like here. Yeah, you see here, for example, these two helices are stacked on top of each other. Here you have a little loop, you have a non-Watson-Crick base pairs, Watson-Crick Uxtin or whatever, and these adenines are interacting in the minor groove of this one, maintaining this coaxial stack, and the third, pushing the third helix on top so that these two can talk to each other later on. All right, so that's why I think we need to annotate these non-Watson-Crick base pairs and give them names. And so we in a nucleotide, we recognize the Watson-Crick edge, that is the usual edges. And you have the Uxtin edge, that is with N7 here, or C5 in pyrimidines. And that's the name of the crystallographer who got first crystal structures of with this type of base pairs. And we have the sugar edge, because the O2 prime is what makes the distinction between RNA and DNA, and they're always in flight in many base pairs. So you can, all nucleotides, you can simplify where the triangle, and then you can look at all the possibility of mixing these triangles, and you have all the families of base pairing, except that the sugars is asymmetrically differently disposed with respect to the base. And so you can either have the sugars interacting with the base and they are on the same side of the interaction right, or you are on the opposite side, like here, and this is what we call in trans, okay, like and so that multiplied by 2 the number of possibilities. And in the end, you have this type of figures with 12 families. They all exist in 3D structures. So helices, DNA helices or RNA helices, only this type of Watson-Crick in cis. All right? All the others, they are important for 3D structures. And without them, it's impossible to fold any complex molecule. Right. And so, for example, in blue area, these are very, very common discretes because why they are anti-parallel, like in this one, so they fit together. They are internal hairpin loops. These will be occurring in triple helices. That is when the third strand comes interact with the RNA. And the last one is the backbone term, when that's why they are parallel with each other. So it's a single strand folded back on itself. Sometimes the strands will be parallel with each other. And you need base pairs to maintain this type of contacts. All right, so now just to emphasize the very important point here, here you have the 2D structure, standard cloverleaf of tRNA, and the Leavitt base pair is here somewhere. That is 15 for 48. He discovered it from couple of sequences in 69, you know. He identified wrongly, but still. And now the way it is stacked together, you see all these type of non-Watson-Crick base pairs linking them together in the core of the tRNA. What is really important is that we have many, many sequences in RNA. And so we can align this from this structure. You can align all tRNA structures very precisely so that you know that this one is always base pair with this just part, these two parts are base pair together, these base are base pair together, etc. And that's an experimental structure. We can from the experimental structure, you can then align all sequences and you can see how they behave through evolution. And so an accurate model should explain the conservation. You see here, the U is always conserved. Here you see the U is always conserved. And sometimes it's semi-confirmed, there is a semi-conservation. It's not always there. So if your model is right, you should be able to understand sequence alignments. You should be able to produce a sequence alignment that is really the reflection of all these families of homologous molecules, right? And this is a very important point, I think. Now, if to simplify this, here in yellow, that's the 2D structure. In red, that is the 3D structures. That is those that maintain the structure. But you have to find them. And we know that they interact with each other here, or they interact with this one, they interact inside them. Without those, there is no tRNA structure. It will unfold, all right? It will not even fold at all. All right. So that's why we introduced the notion of interaction networks. So it's a good prediction, it's a correct interaction network. So when we look at an RNA molecules, we not only look at the Watson-Crick pairs that you see, the GC, UA, etc., GU, but we also look at the non-Watson pairs with these symbols that we introduce here. All right. But in addition, we use a stacking, that is whether this helix is stacked on that one or this helix is stacked on that way. And this is really critical. And you can measure all these parameters, and this is included in INF, interaction network fidelity. That here, that is really the Matthews coefficients here with true positive and false positive, etc. And so you can do that for inf Watson-Crick, inf non-Watson-Crick, inf stacking, and you can bring them all together. All right, so we have a good idea of how good these parameters are respected. So RNA puzzle is a collective blind experiments for the very automatic, so it's really equivalent to Cas for protein. So we need RNA structuralists and volunteers of RNA modelers. And the main difference with between we started 12 years ago, and now is Teach Amiao in Guangzhou. We continue this. The main difference with Cas is that we accept puzzles continuously on a rolling basis, because not too many. There are less structural biologists on RNA, now it's increasing, so they do not come very regularly. If you have to wait every 2 years, we'll have 1 or 2, etc. So in this case, anytime there is a new one we can go at it. And we have an agreement with CAS that during the period of the CAS competitions, we transfer the RNA puzzles to CASP. All right, so some correlations between the different metrics. You are very good at this LDTT, GTTT, DS, etc. All these very complex parameters, so we can compute them too. But here we have inf all, what is with LDTT. You see that is the correcting is pretty good. All right, also then the inf all with TM is also not too bad. But you see the clash score, it goes up to 900, or 450. So that is dramatic. I don't understand that is in 2024 we still get such bad clash score. We should really not look at this type of models. So here we stop the clash score at 100, and the plot are even naturally a little bit better inf all LDTT, so inf all and TM, you see here, naturally, and the clash score is another problem, right? So now we try to look at some more details. So I look at it's easier with RMSD. It gives you an idea because I know that an helix is 23 angstrom wide. All right. So here you have RMSD going to 250. You see out there. All right. So the here is 100. That is 60. All right. This is a huge number, and these things do not look at all actually. So I think we made the cutoff at 60. And here, that is with the TM, you see you have many things below 0.4. So, but if we remove them, naturally, all this we can forget. All right, so here is with 60, and you have all the slight increase. And what will be very important is that all the large non-coding RNAs are in this section. So we separate template without template, and prediction with template, that is 30, the blue line 30 airstream, which is starting below 30 CR and in decent ranges, and you see that the minimum are really very pretty good. And here this is, for example, the all large non-cloning RNAs, gold, rule, all is very, very important ones. And you see that naturally we are all beyond 30 angstrom RMSD. So just show two examples. This is R1296. This is the HIV RE element, and you see that for 81 was down there, but everybody was the same. Oh, yeah. They all got the same stuff. It's really amazing, very homogeneous. Everyone is over there. This 304, that is alpha 43, it's like the others, no difference. And naturally, that is the R1296 is shortened version, and there is this was published. That is the model from 481, 3.4 angstrom. That is really very decent, very nice modeling, but there is some previous data. This is a very complex one, non-coding RNA. That is a huge one. That is a very large model. And the best one was for 063, 31 angstrom. I don't see why I don't see the plot here. Sorry, but it's down there. So it's most of everybody is over here. All right. And if you just to emphasize this importance of non-watching pairs, you see in the model, in the structure, you have this helix here, this complex helix with non-Watson Crick base pairs, etc. And you have this loop, and you see how nicely it interacts with this green one. And interacts in the minor groove of this deformed helix. Now, in the model, the best model, they are 35 angstrom apart. So they don't talk to each other at all. So that means they totally miss the fold of the RNA, right? Because this you want to know, because that is conserved. If you make an alignment, you will see that these adenines are conserved, that the interactions are conserved, that they can be respected for the full alignment. Okay, so global fold is not sufficient sometimes to capture the key 3D features that maintain the architecture and explain the biological function. We want to explain biological function. We want to help experimentalists so that they know which mutation to make to prove that their enzymatic function catalysis is really this residue and not that residue, etc. All right. So, gosh, why did this disappear? Large ribozyme, all my plots disappear. This is okay. It's large ribozymes, they are all over there. You see, but what is really amazing is that some were very good and some were all over the place. I don't understand. So That is also a PC to Mac problem. Probably, gosh, okay. It's good. Many, that is homology model is very different, so it's different story. All right, now, so to rank, if I do the RMSD, so we plot the same kind of plots that Rachel showed. Here are the teams. Here are the number of submitted targets. So we have 34, the total number. So most of them did produce 34 targets. The number of targets in the best 5. So we take the best 5 and number how many times they are in the best 5. And then you have the score here. You see 49, 34. The maximum will be 170. So that means with 49, 50, you are below the 1/3. Right? So it's still a big margin to be really the best. Right? But still, you can see this one there. That is just your RMD. Now, what's important? 38 groups, 27 groups are never in the top 5. See? And 38 were at least once in the top 5. Okay? So many groups are very far away. So RMSD with template is the same story a little bit there. Here we separate with RMSD without template. The arrow shows AF3 folder server is down there, all right. So, and the others are indicated. And we use also the Z-score. That is the Z-score that Riju and Rachel introduced in class 15. So we kept that one for continuity. And here you have the Z-score. This is again AlphaFold 3. And here you see Z-score without template. AlphaFold 3 is number 1. But is 19, the number of points. And the number 2 is 416, 17. It's exactly the same point. It's exactly is the same. And here the first two are also almost the same, 29, 28. Here 35, 32. All right. But here with template 3 or 4 is very low. I don't understand. So it's strange that AF3 did better Z-score without template. It's good. So now I can look at all the various scores that we have here, and I put here the team, and you see that 481 is very frequently there. 183 is very frequently there, and you have some other ones there. And so in the end, if I go there, if I count, the first 2 positions, 481 comes 7 times, 6 times is first. 183 comes 5 times, 5 times second, and 63 comes, etc. So I think that exactly like with Rachel, the first 2 are clearly 481 and 183. And this is TJ Chen, and this is Lin Yuan in Guangzhou, and this is the other, and 304 is AlphaFold 3. Okay, so to conclude, some general remarks. So there is no single comprehensive scoring function for assessing accuracy. We need more, and we think we should develop new ones for understanding better or scoring better. Some scores are local or more global. More are based on atomic coordinates with a consideration of interresidual contacts. INF is the only one where base pairing is assessed directly, and we need to know which kind of base pairing is occurring. There is no single prediction method that stands out yet. We saw the differences. The successful ones are knowledge-based, so in human experts, and use various specific force fields, statistical models. They encode certain RNA structure folding rules and allow for intervention of human experts. That's the important point. And success will still depends on the type of puzzles or the type of target and on the human expertise. So you can never tell which one and who will succeed. And so the future was just quickly a combination of current methods with the powerful AI generalizing folding rules extracted from large datasets, but the systematic use of multiple sequence alignments would lead to a more evolution-based modeling. Sequence alignments is not only a MSA just to retract to obtain the 2D structure. You will have to extract also 3D elements from sequence alignments that are properly done. And this is not necessarily easy and straightforward. But we have many examples now to try to find the roots. So again, double 4 is not sufficient. I told you the non-logic pairs, RNA modules, all these are not there. Problematic structural features is clearly coordinated interfaces and ligand binding, handling ions, with clash scores. surprising. Impact of external uncertainty, low quality or poorly resolved cryo-EM. This is sometimes also difficult to because our tools are atomic based, and so if the bases are everywhere in space, it's very difficult to assess the quality. And so now issues and questions. I think we should be really distinguish clearly the best method for fitting with accuracy into cryo-EM mat. You have a cryo-EM mat and you want a tool to fit correctly in there. This is a very important problem. But this problem is distinct from the best method for which you model without experimental data. So we should keep these two, this assessment separate. And so we have also homologous structures like group 1, group 2 introns. This is very important to be able to model in a homologous structure, but this process is complex and it's not the same as predicting de novo structure. And we should keep them separate also, because it's not easy to model a group 2, a variant of a group 2 intron that is more than 400 nucleotides, all making non-Watts split base pairs. If you want that correct, it's not necessarily straightforward. And also what should achieve. So as Rachel said, with template, the accuracy is all right. Also, the acids are going large, spreading the results. Without template, the outcome is poor and very irregular. But I must say the targets were very ambitious. 400 nucleotides is really amazing. That's too ambitious. Ole or gold or etc. That's really massive RNA. The distinction between template versus non-template is central to the prediction methodology and to the final assessments. And this is not easy because we have to see whether you have lots of sequence homology, is a structural homology, how do you compare them, how to assess them, how to include them. And this distinction, I think, is not easy, and to keep it clear, and especially with AI method, where if you have seen one tRNA, you have seen all tRNAs, right? And so we may need some clarifications there to how to template versus non-template with AI in mind. And so now I think I come to the end, and I would like naturally to thank Chichao Miao over the years working on this project, and Fan Bu is a PhD student, also working in Guangzhou National, and she did all the calculations behind the scenes, etc. And if you have any puzzles, sees the org file. Thank you very much for your attention.